And today's first talk will be given by Meghana Missula, and this talk will be on automated inference of production rules for glycans. So, Meghna, if you're ready, you can start, put it in presentation mode, and start.
Uh, it's visible?
Yeah, we can see it.
Great. Good morning, everybody. I'm Meghna Missula, a final year computer science undergraduate student at IIT Bombay. This talk is about automated inference of production rules for glycans. It is to be presented at CMSP 21. My collaborators for this paper are Professor Ashutosh Gupta from IIT Bombay and biologists Mr. Anshuman Vishwas and Mr. Mukund Thattai from the National Center for Biological Sciences at Bangalore. So, we all know that there are many computationally hard problems to solve in biology. Some of the very popular examples are determining the future mutations of cancer cells, developing brain models, and so on. And from our knowledge, we also know that formal methods are known by nature are known to solve computationally hard problems. So in this talk, I'll be presenting a novel application of formal methods in biology, particularly inference of glycan production rules. So before getting into what are glycans, what's their story, etc., I'll be talking about why at all are we considering this problem? Why is it important? So, glycan molecules help us identify cell types in organisms, like how IP addresses are used to identify computers or computer networks in a computer science setting. When biologists or chemists develop any new drug, they must ensure that it doesn't meddle with the processes of a glycan, otherwise, it could lead to serious complications. So now that we know that, okay, glycans are not something to be messed with, we know they are important things, but biology still has questions about how they are produced, as carrying out research in this is expensive and it happens limitedly. So when we looked at the problem, we thought, maybe formal methods can help us in solving this problem. So let's try and understand how exactly can we model this. The assembly process. So the ability to control the assembly of small building blocks into large structures is of fundamental importance in biology and engineering. So familiar examples in this are synthesizing DNA from nucleotides and linear proteins from amino acids. And in both these examples, the synthesis is templated, which means that the new DNA or the protein molecule is essentially copied from an existing molecule. What we are dealing with in this paper is also essentially an assembly process. We have seen why glycans are important and that they are made through an assembly process. But what exactly is a glycan? So, glycans are tree like polymers made up of sugar monomers, building blocks. They are found on the surface of all living cells, and distinct glycan trees act as identity markers for distinct cell types. Simply put, glycans are branched sugar oligomers covalently attached to proteins on the surface of all living cells. In the figure, you can see there are different shapes and colors which represent individual monomers, and the tree itself makes for a single glycan molecule. Enzymes. Enzymes are proteins that assemble glycans through successive additions. This process is called glycosylation. In this illustrative figure, we can see the details of glycan production. A glycan is a tree-like sugar that has nodes linked by edges. It is attached to a substrate protein at the root, which has been labeled R. Curved boxes in the figure represent compartments within cells, which are the sites of glycan production. Each step of glycan growth, the black arrows, represent the addition of a single new monomer to a specific attachment point on the tree. And each such step is catalyzed by an enzyme, as we can see this E1, E2, E3. So at any stage of growth, the tree can either exit the chosen compartment or it can move to the next compartment. Uh, what we can also observe is that these enzymes are known to act only at particular points. It cannot happen that any enzyme can act at any substrate glycan. So there is some kind of a specificity involved. Moving on to what are the properties of this glycosylation. One, as we already have seen, is specificity. The monomer attachment shown in the previous slide happens at a specific point in the tree, like how a puzzle piece can fit into only a particular point of the whole puzzle. There's also inter and intra-cell variability. So intra is to get the glycans using as few enzymes as possible. All biological processes happen with limited resources. And intercell variability is something that is created by glycans themselves as they identify the cell type. And in the case of glycans, enzymes operate with micro heterogeneity, meaning that a given site on a given type of protein is associated with multiple types of oligomers in stochastic nature. That is, oligomer grows one monomer at a time as the protein moves to successive compartments, stochastically encountering various enzymes within each compartment. However, when we look at the glycan profiles of individual proteins, they are narrow and reproducible. Now, the natural question is how can this stochastic and heterogeneous biosynthetic process generate narrow and reproducible glycan profiles? And hopefully, our research can help in answering some of these questions. In all the assembly processes, the key problem is to find the building blocks making up the target, which is like an inverse problem in itself. And the assembly process of glycans can be compared to the theoretical framework of Van tiles, which are square building blocks with colored sides that stick to one another along sides with matching colors. For efficient assembly of Van tiles, their coloring must be cleverly chosen, so the target shape is the unique output of the stochastic assembly process, which is also the case with glycans because the assembly happens in a specific manner, like how colors have to be matched here. There, some substrate and the monomer has to have a matching for the attachment to happen. Now, before I shift from the biological motivation to the mathematical modeling of the problem, I'd like to pause here and see if there are any questions.
I don't think there are, so you can continue.
Okay. Okay, if there are none. Now I'll jump right into what is exactly the problem that we are trying to solve here. Glycans, tree-like polymers determined by cell type, both of which we've already seen. And there are production rules governing their complex assembly process because of specificity, stochasticity, etc. And if we look at these production rules as a program and glycans as their output, the problem is to basically find a program that produces this output, that is, find the production rules which can give these glycan molecules as output. And this is like any other program synthesis problem now. When biologists try to infer these production rules, the process is manual. It uses prior knowledge and scientific observations. There is also a previous paper by one of my collaborators in which they developed algorithmic constructs to infer these rules. However, they're valid in only specific situations and they do not take into account for the stochastic nature. So the key question here is that can we do better using either formal methods or other techniques? Can we do better than this? Now, a theoretical calculation by my team found that in an average setting, there could be about 10 to the power 70 rule sets possible. And here, because of this the large computation, formal methods sets in. And there is a clear need for automation to find these rules involved in the synthetic process. Our method for production rule synthesis. So our rule synthesis method is the first of its kind. It is the first automated synthesis method for the production rules and which looks at the computational problem. Our method of synthesis is similar to counterexample guided inductive synthesis. Several methods for solving problems of searching in a complex combinatorial space use templates to define and limit their search space, such as learning invariants of programs and synthesizing missing components in programs. We also use templates to model the production rules. Our method is iterative. We first construct constraints encoding that a set of unknown rules defined by templates can assemble the input set of molecules. The generated constraints then involve Boolean variables, finite range variables, and integer ordering constraints. We solve these constraints using an SMT solver. We call the query the solver synthesis query. If the constraints are unsatisfiable, there are no production rules for the search space defined by the templates. Otherwise, a solution given by the constraints is returned. We call the query to the solver as synthesis query. If the constraints are unsatisfiable, there are no production rules with the search space defined by the templates. Otherwise, a solution of the constraints gives a set of rules. However, there's also an additional requirement that a molecule which is not in the input set should not be produced by the synthesized rules. Therefore, the method looks for producible molecules that are not in the input set. If again, this is also assisted by templates. And we again generate the set of constraints for the unknown molecule. We again solve these constraints using the solver, which is called the counter example query. If there is no such query, our method just reports the synthesized rules. Furthermore, there are many variations in this problem in the type and quality of data given to us. I'll briefly talk about them later. All of this is implemented in our tool glycans. So far, we've looked at the uh, uh, biological context and the need for formal modeling. Now, I'll jump into the formal model. So let's do this through an example. Let's consider there are two glycan molecules given to us. The only difference being that their left and right child are interchanged. These are the input glycan molecules. Now, if we were to synthesize, and yeah, the set of monomers in this are only three, the red, green, and the orange circles. Now, intuitively, if we think about it, we can see there are four rules which make up these two molecules, the four rules displayed on the slide. And basically, the green and the oranges can combine together to give us the input molecules. But if we look closely, can these rules actually produce molecules which were not there in the set? Well, yes, because the orange ones could combine together to give us a molecule. The green ones could combine again, which gives us extra molecules which were initially not in the input set. So let's see what the correct rules are once we've seen the algorithm. Input and output to the algorithm. So first input is the glycan molecules themselves modeled as trees. And then we have maximum rule depth. We do not want unbounded rules to be produced, and to basically make the problem finite, we add a new parameter which limits the rule depth. Then we have number of rules to be synthesized, and the output of the problem is the synthesized rules themselves, if they exist. Let's see at a high level picture of the algorithm. First, we initialize the constraints that basically encode these production rules. Then we check for if there is a satisfiable model with these constraints. If yes, if we do get a model, then we get the rules by the model, which are given by the model. And if there are not, then it just throws an error that okay, there's no rules that the algorithm can find for these constraints, and maybe we have to change some parameters to get rules again. Now, here's the catch. So we have rules from the satisfiable model. Although these rules, like we've seen in the previous example, they can give us a molecule which is not there in the input set. So we go ahead, we check if there's an extra molecule that can be produced with these rules. If that is the case, we add new constraints to encode that we do not get this counter example in the next iteration, and the loop continues. If that's not the case, well, we have found our rules, and this is synthesized rules are returned. Now let's look at our previous example again. We have these two input molecules. We have to synthesize four rules because we were looking at four patterns. And the rule depth is 2, the root itself and its child. The algorithm returns these four rules as the synthesized rules. And if we have a closer look, we can see that this never actually produces an extra molecule, because whenever there is green in both the sides of the tree, left and right subtrees, what we can see is that the orange blocks one of them. The orange is present in one substrate, so essentially both the greens cannot act at the red color root at the same time. So this prevents from creating extra molecules. Now, in this case, we might have manually calculated this, or just by looking and from our experience, but when the scale of the problem increases, it might be really difficult to solve this, which is why we our algorithm basically helps. Uh, right. So there are also many variations to this synthesis problem. I'd like to briefly talk about a few. There are fast and slow reactions. Every chemical reaction has a rate. Slow reactions are overshadowed by fast reactions. What fast reactions do is that basically, if there's a partial molecule in a compartment, the reactions happen rapidly. They finish the molecule, and only then it gets passed down to the next compartment. Runaway reactions are basically repeated applications of a particular subtree. And so a negative molecule, which was initially considered an extra molecule, will cease to be negative if it only has a repeated application of a particular subtree of an input molecule. So we do not consider them negative anymore because runaway reactions happen in cells all the time. Then there are compartments, which we have already seen. And there could be incomplete and noisy data, which means the data given to us might not be completely accurate. So we model for some possible deviation from the input molecules, and if the negative molecules are within a few monomers of difference from the input molecules, we do not consider them negative. In the interest of time, I had a few slides on demonstrating why fast and slow reactions are helpful. In the interest of time, I'll skip these. Coming to our tool, it's written in C++ using Z3. The data has been from the biological literature. It is an open source, publicly available tool on GitHub. Feel free to check it out. Coming to the results, these are the results from real data sets. D4 human chronoid gonadotropin from cancer cell line was a motivating example. We also tested it on synthetic data sets, and what we can observe here is that changing parameters can change the time that the solver takes to solve the problem drastically. And in some cases, like the last row in D4, it might not even be successful in finding the rules because either too few rules were asked for, or the rule depth was small, or it could be any reason like that. So in D1, even by reducing the first two parameters, what we can see is that we can synthesize rules, but taking a longer time. And then giving an extra compartment did not affect the performance. We also observe the trade-off between number of compartments and the depth of the rules in the second and third row of D1. For D2, which is the example with runaway reactions, we synthesize it with 7.97 seconds. And we also learn large rules, which is that they're adding many nodes at a single time. So the main inference is that if we give too many resources or too little as input, then the tool runs for a longer time, as the search in combinatorial space is highly sensitive to parameters. Well, the next steps are to interpret these results that we get from the algorithm as output and validate them from the biological experiments. We also need to check the viability of the solutions and their possible potential applications to other fields of biology. And there could be more variations of the synthesis problem, like the compartments that we have currently modeled have rigid boundaries. We could change them to have flexible ones. There could be stay models, which means that how much time does a molecule spend in a compartment is fixed. And a theoretical possibility is to consider this whole synthesis problem as a minimization of a modified version of tree automaton. And conclusively, so in this talk, we have seen what glycans are, what the process, the assembly process is, how have we modeled it, what is the algorithm, why do we need the algorithm specifically. In the paper, you can find more details on the algorithms themselves, the formal justifications, and more experiments regarding the variations of the problem, and so on. Impact necessary to discuss what is the impact that this is creating. So once we have the data, when we look at it, we analyze it, we can find that our initial question, why is this stochastic and heterogeneous process still creating narrow profiles? We can understand and answer some of these questions, and we can apply this to other real glycan data sets. And we have also opened the table for other people to apply the same concept in different aspects of biology. And hopefully, this project can create that difference. Thank you. Yeah, that's it. If there are any questions, I'll be happy to take them.
Yeah, thank you, Meghna, for the nice talk. Radhakrishnan Delhi Babu has a question.
Yeah, I have a question. So, nice talks, first of all. And see, your method is full abstract. Okay, the main thing is you make a glycan with input and do some process and get some results. You said that the four benchmark rules. You said that, but when you go for the final results, you make a six rules and eight rules. So you said that four rules, but now changing for different rules. So can you do any permutation combination for different rules? Can you go back the slides?
Sure. We synthesized four rules.
Yeah, four rules. No, the results. Yeah.
Yeah, here.
So the four rules fixed. Okay.
Right.
Because I see that your paper some equation, because I didn't understand that, but I see that equation. But the equation says six rules you gave. So when you say that in the results you say that the number of rules is number of rules 8 and 6 and 7. How the combinations apply? Go to the last slide. Go to the last slide. The rule box slide.
Okay. Okay, the result slide.
Yeah, here. See that number of rules 7, 7, 6. So my question is 4 rules, you say, but how the rules changing here? Any method of changing the rules?
So the rules are specified through an input parameter. We can specify how many rules to create as part of the input. So if I say create 4 rules, it will create 4. If I say create 7 rules, it might create 7 or lesser than 7 if there are less than that.
No. My question is 7 means because if you say only 4 rules, but how will you come from 7? So the combination of 4 rules, 7 rules?
This is a different example, sir.
Okay. Sorry for that. Okay.
Yeah.
Because I didn't get the whole. So the second question is, the glycan, whatever the synthesis is, I think the new. I'm pretty sure that. So did you apply any logical things here? That means you apply in data log or prolog, implement and get the results in logical programming kind of things.
So we validated them with our biologists on this project that do these rules make sense? Do these rules actually, are they something that you also thought about when you looked at the data set?
Okay. So may
I answer this question? Yeah. Hello?
Yeah.
Hello?
Yeah.
So the thing is, you simply
Ashutosh, your voice is breaking.
Hello, sorry. Can you hear me now?
Yeah.
Yeah. So this is essentially a program synthesis problem where rules are basically programs which you need to figure out which produce exactly set of trees. Okay. Now the question is there a simpler way to synthesize them? For example, you're suggesting maybe there is. So we tried one, but it does not appear to be us because the some of the example is fairly tricky, and the solutions are like you will see that it can be solved using 6 rules, but there are no 5 rules which can produce this set of molecules. So this is very, very heterogeneous environment, and this is like very much like program synthesis, where simple solutions won't work. You need to go and use the full hammer of Fromm's method. I hope that answered the question.
Yeah, sure. So then I can ask one more question, Suprit. Maybe time is there.
Yeah, go ahead. I don't see.
So the interesting thing is if you get this result to compare with biological lab, and you can find out that both are equal. I think this is one of the great achievement. And see that because the main thing is if you say open question, the smelling. So basically the smell also same, the glycan, how the working the same thing working on smell. So how the small organ go inside, how this most of things, not only smell, you can take a tablet also, you can take anything in the inside the body, how the reactions can happen. So everything can be synthesized in future. So I think this is a good way. Nice work, and then thanks.
Okay, thank you.
Right.
Are there any other questions?
Yeah, I have Venki here. I have just one question. Yeah.
Go ahead.
Can you go back to the So here your rule depth maximum is 4. Is it that in biology we find only rules of depth 4, or have you restricted yourself to 4?
So, like I said, it could take a very long time if we give it a very large parameter. And generally, it has been observed that rules are generally maximum of size 3 or 4. So, generally, we try these numbers 2, 3, 4, and then we see how it performs. Yeah, that's from the domain knowledge completely.
Yeah, so maybe I can also add on that. So so the what happens is that so what is depth? Depth means that the protein has to look at the recognize a bigger portion of the molecule that it has already been built. So what else I should do? What should I add? Okay. So more you have to recognize, more work enzyme has to do, more basically it's a lot more work for cell uh, to learn how to recognize this pattern. So nature has a cost associated with the bigger rules. Okay, so it is expected these depth are small, number of rules are small. This is all our assumption we are working with, and the biological knowledge is limited, and somewhat actually domain knowledge, as Meghna has said, tells us it should not be more than 3 and 4, but there is no proof for it or biological evidence, hard and fast evidence is out there.
Yeah, for example, if it's a long glycan, you may have a rule that the first and the last monomer must be something.
Yes.
And
yes.
that
That's
you
right.
will consider as a rule depth of more than 1?
Yeah. So you are asking us what is our language of rules, right? So currently we have a language of rules that is portion of the tree has to be recognized existing there, but it can be arbitrarily complex as you suggested, right? It can be far-reaching. You're looking at the like my three ancestor app should be the red one, okay? But I don't care what happens in the middle, right? It makes arbitrarily complex rules, right? So and there sometimes the rules are like this, ki at this node there should be nothing. If there is something, then I cannot be applied, okay? So this becomes a non-monotonic kind of rules, okay, which you cannot really monotonically learn, which like for example data log somebody suggested, right? So yes, those rules are possible, and our language we support them. But however, it just as you allow more and more bigger and bigger such space, it gets more and more expensive.
Yeah, sure.
Okay. So in the interest of time, let us move ahead. So thanks, Meghana, for the nice talk.
One question in the chat box.
Yeah, there is from Alvin. Okay. So Alvin, do you want to go ahead and speak up?
Hello.
Or maybe I can, Alvin.
Hello.
Okay, maybe let me read it out from the chat. Yeah, okay, go ahead.
Yeah, I read the question. What is isomers in this context?
Does the rules distinguish isomers?
No, okay. So if you're saying the labels only they are different, like just if structurally they are similar, then it doesn't. You can give any names to the monomers, and if structurally they look similar and the So the arrangement and the combination is the same. It doesn't distinguish. Yeah.
Okay. Okay. Thank you, Meghna.